Okay, it's 6 p.m. We'll wait 1 or 2 more minutes just in case with a few people. So thank you for being here. Okay, perfect. Where is it? Can everyone see my screen? Everyone can hear me well?
Yeah, yeah, very good.
Perfect. So we're going to start. Thank you very much, everyone, for being here today to this webinar, focusing on harnessing artificial intelligence to unlock new frontiers in biotechnology. So I'm pretty sure you have heard or read similar headlines in the recent years. So AI is on everyone's lips, and actually AI technology is becoming more present in our daily lives. So when we use our phones, you can find a photo being classified for you. When you use social media, face recognition, when you talk to your device. So some industries rely as well on AI with voice recognition, for example, and science is not exempt to that. We've seen major change. Now that we hear all about it, actually to know about opportunities, challenge, what to look for, we believe it's really important to actually talk to experts in the field. So this is why we are here today. And it is my pleasure to be joined by Dr. David Nelson from the New York University of Abu Dhabi in the United Arab Emirates, Dr. Min Zhao from the University of the Sunshine Coast in Australia, and Dr. Roy Costilla from the Cawthron Institute, so in New Zealand. Myself, I'm Max Plouvier, an algae biotechnologist at the Cawthron Institute as well. And just to give you a bit of background information, the Cawthron Institute is the New Zealand's largest independent science organization that focus on preserving New Zealand's unique environment, contributing to its economic growth, and also tackling global issues. So therefore, our science is really broad, from ocean health initiatives to aquaculture efforts. And as you can imagine, AI is going to play a big part into how we do our science, and it's going to help us as well, to generate and improve some of those fields of research or industries. So I would like to acknowledge and thank the Cawthron Institute, the University of the Sunshine Coast, and New York University in Abu Dhabi to supporting this webinar by allowing their staff to present today. And in addition of being a Cawthron staff, I'm also the president of the Australia-New Zealand Marine Biotechnology Society. So I would like to acknowledge and thank the society for supporting this webinar, and as well as members that have helped. So Yaming, Elizabeth, and Min, thank you. Thank you for helping organizing this webinar. And just to provide you some background information about our society, we are relatively small, about 200 members, and we are promoting marine biotechnology across Australasia. So, as the name indicates, mostly Australia and New Zealand. And our mission is to foster collaboration, innovation, from fundamental research to real-world application. And we are trying to bring key players in the field together to promote responsible and impactful marine biotechnology across the region. So as I said, please go check us out, and I will take the opportunity as well to promote, our conference that is going to happen later this year. So we are trying to promote marine biotech via newsletter with our memberships, via webinars like the event of today, or conferences. So if you are interested, please join us. I mean, we are aware that the war situation does not make it really easy at the moment, but it would be a pleasure meeting you later this year. So now before we have great talks lined up and before going into the depth of all those talks, I just prepared a few slides to, give us some background information about AI. And so we hear all about it and we hear about different terms. So AI, artificial intelligence, you may have also encountered the term ML, machine learning, or deep learning. So sometimes it can be confusing. Um, and overall, AI, artificial intelligence, is the broad concept of intelligent machines. So it's training machines to mimic some specific human cognitive function like problem solving and learning. So AI can be predictive or generative. So now we've all used some tools available to us to generate some content. The figures, the images that were shown and that are shown today were actually generated by AI. So what a great tool for that. Obviously, you need to check because it does like to put some tiny mistakes in the content. But when it's you can work with the AI to design what you want. And so machine learning is actually a subset of AI, and deep learning is a subfield of machine learning. And machine learning and deep learning algorithms are trained to generate model for pattern recognition and prediction. Actually, those algorithms are really good at finding subtle correlation or hidden trends in data that allow us, to go in depth inside, use the data to go in depth and understand new things. And the difference between machine learning and deep learning is their structure and how they learn. While machine learning requires some human guidance, for example, when you need to label the data, deep learning with its depth, we have the use of artificial neural network. It can automate feature extraction that will on its own, and then will be able to either recognize specific pattern or predict specific data. And what I find really interesting about artificial intelligence at the moment is that, we see it pretty much daily. And we may believe it's actually really new, but it's not true. Actually, AI was born more than 70 years ago when key scientists were interested into developing artificial intelligence in 1955. And this is what they propose. So the study of AI is to proceed on the basis of a conjecture that every aspect of learning or any other features of intelligence can in principle be so precisely described that a machine can be made to simulate it. An attempt will be made to find how to make machines use language, form abstraction and concept, solve kind of problem not reserved for humans, and improve themselves. So really interested to see, that they were thinking about those concepts more than 70 years ago. However, little ambitious of them, wanted to solve some of those gaps, over a summer. But anyway, it's really good to see where we are today. And actually, this graph will summarize really well where we are. It's specifically focusing on the use and the development of artificial intelligence for biotechnology and applied microbiology. But all the field of research would have, a similar trend. And what you can see is a surge in the number of publication, being submitted and published in the field after the year 2010. And actually, what has led to this surge is, in addition of interest, is the development of computational power and as well the availability of large data sets that are needed to fed into the algorithm. To find the patterns that I mentioned before. But another change is actually the fact that now data sets are becoming increasingly available and they are publicly available. So this makes it really useful for research in the field to improve because researchers can access this data. And now I believe we're entering a new era with the development of agentic AI, so AI that can do more tasks on their own. And just to provide an example already of some of the breakthroughs that has been achieved with AI and the amazing development of the technology is, for example, AlphaFold, which is a deep learning algorithm that allow you to model protein structure really accurately from a one-dimensional amino acid sequence. And so now actually we have 200 millions of predicted protein structure available in databases. And this is really useful for structural biology, but also biology on its own. So to study protein and to provide insights into their functions and interaction. So this led to a Nobel Prize in chemistry a few years ago, and AlphaFold is still developing. And now it is possible to actually model multimers and to study protein-protein interaction and protein-ligand interaction as well. So obviously, this provided a big leap forward in biotech, and many industries and scientists are now using this tool for drug discovery, disease, the identification of biomarkers, and also enzyme engineering. So truly evidence, the potential of the technology. But as everything else, with opportunities come and new things comes advantage, but inconvenient and challenges. So although today I do not want to enter a debate about the potential risk of AI, the potential environmental impact, and the need for legislation, I just provided a few key papers that I found in the field, and pretty much it is pointed out by those researchers that it is critical that AI is explainable, and that there is the need for methods and process that make the decision making of AI system transparent, interpretable, and trustworthy for humans. So to understand why a model made a specific prediction, and obviously for also users to know what to look for to really be able to trust the data. And actually, in this document, is AI overhyped? A key scientist, have clearly stated the need and the importance to maintain humans in the loop, obviously to prevent issues down the track and for obviously losing control. And therefore, humans are still needed. And I had a little play about, using Copilot, and I thought this figure would summarize pretty well, what is what is being discussed, and took a few iterations. But at the end of the day, we still need humans' interaction to work, with AI to make it, really successful. But now, jokes aside, as I said, AI is already changing, many industries, changing science, the way we do science, but it's also going to have, a significant impact in marine biotechnology. So marine biotechnology is the study and use of marine organisms and their compounds for biological progress and human benefits, so to develop product, bioprocess, and technologies. Obviously, the impact of marine biotech is really broad, and it's an important field because the marine environment is so rich that there is really the potential for new discoveries. And to illustrate that, actually, a document, Marine Natural Product, which is a review published recently, they, in this review, stated that around 1,200 novel compounds have been found in the year 2024 from the marine environment. And this was 1,000 more from the year before. So really just to show you the richness and the potential that we have, in studying the marine environment and the potential for marine biotechnology. And just to provide an example about, how AI can help in marine biotech, I will take the example of my own research, microalgal biotechnology, where we see AI, now being implemented with the use of various sensors, various imagery system, for real-time monitoring of the culture, for species identification, and this obviously combining AI modeling with those various techniques can allow for the optimization of the growth conditions of the culture. And the ultimate goal would be AI-guided bioprocess control. Therefore, you're able to supply the right amount of nutrients and lights, for example, while increasing productivity of biomass or either a specific compound. So therefore, you increase your profit while reducing your cost. There is a few examples of the use of AI in marine biotech that we are going to discuss today. But now we'll take the opportunity actually to introduce Dr. Min Zhao, that is going to talk to us about a system that he has developed to actually democratizing the use of AI. And Dr. Min Zhao specialized overall in multi-omics and the development of AI to democratize its use in bioinformatics. He will present to us Smart Blast, an AI agent that interprets research goals, design appropriate search strategies, and evaluate whether results meet the researcher's objective when using Blast. So Blast being a bioinformatic tool to study, for example, the evolution in a gene between organisms. So his session demonstrates how AI can make complex tools more intuitive and goal-driven. Thank you, Min. The floor is yours. I will stop
Thank you
sharing.
so much. Thank you for the introduction. Thank you for organizing this, Max. Let me
No
share my
problem.
screen. So all good. So Max, you guys can see my screen here? Hi, Max. Can you see my screen?
Yeah, it's all good.
All good. Good. Let me try. Okay. Because sometimes I'm doing the full screen, I was totally lost what's happening inside of Zoom. Anyway, let me do in the full screen first. Thank you so much for the introduction. So today my talk is this is a new project. Still not finished yet, but it's kind of the new ideas. I mean, just a quick idea, probably a few months before. I called it a Smart Blast. It's an AI-driven sequence discovery for the marine biotechnology. And so the basic thing is we are going to going through. So what's happened? So like the Max, you have been introduced a lot of the data have been have been accumulated for the marine genomics. I will give some of the examples and I will share what's the motivation for me to developing the Smart Blast and what kind of feature we have and how can we reuse some of the features. So it's very beginning. I probably just a very broad introductions for what's happened inside of marine genomics. Um, inside, I mean, maybe starting from 2000, from 2000 when we finished 2000 and 2001 when we finished the human genome, the genomics data is expanding quickly like in our marine genomics field, so huge number of the genomics, transcriptome, proteomics, and also the like the metabolic analysis, all those kind of data, they can characterize the cells or marine organisms from different angles. It's huge tons of different data. And in the traditional way, we probably, for different data, we probably use different tools. So for example, genomics data, we will be generating the DNA sequence. Then we need to, first step, we need to do in the genome annotation. Then we need to use the genome annotation tools, like the gene prediction tools, to identify where is the gene regions. And then follow up, we probably need to do in the blast search. We are trying to mapping those kind of the genes from our marine genomics data to something we already know. For example, some of the protein, they have already identified their function in human. And in our marine genomics, they probably have some of the orthologs. We mean the similar protein in the marine genomics. They can play the same function, similar function. So we are going to use hundreds of different tools, bioinformatics tools for different purpose. That makes all the bioinformatics analysis is very challenging. The reason is you need very skilled, or either we say advanced bioinformatician to conduct this kind of analysis, because different tools if you check over like the probably online bioinformatics analysis server like the Galaxy, you will find probably hundreds, thousands of bioinformatics tools available. Pick up the right bioinformatics tools is very challenging. Like we say the gene predictions, probably you can find hundreds of different tools. And which one will be good for you? That's what be kind of things you need to testing a lot of different choice and figure out what's the best one for you. This is very time consuming. Pick up the tools, build up the workflow, so build up the pipeline for for the large scale bioinformatics based analysis. But now we have some of the advantages. So Max, like you have been introduced, generative AI have been helping us doing a lot of the kind of the save us time. I mean, doing a lot of the works in an efficient way. So AI can kind of understanding the background. They can kind of helping us to doing the decision. I know this decision may be not perfect. And now AI, they can also take the actions. So this is the different level for the AI. They are not just generating the text. I mean, you ask a question, they answer your question. Now they can control your computer. They can run in the computational jobs. They can return the results. So this is the general background, and this makes me thinking about how can we use the AI to doing automation for our daily marine genomics analysis. So this is the general things I'm thinking. Just to jump into the algae. So algae is one of the large diverse of the organisms we are focused on. My group we are focused on. I mean, there's huge number of species inside of algae. So we know a lot of the green algae, red algae, but they are diverse of different species with different functions. They have tons of the natural products can be used for different functions, like treating the cancer, fighting with the aging, or probably either way we say the global change. A lot of the things we can find the solution from the algae. Because my research, so because my because our group we have been focused on one type of the red algae we call the asparagopsis, and the asparagopsis can helping to reduce the like the methane emissions from the animals. So this is one of our focus. But because we are working on the bioinformatics, we are not just focused on one group. We are going to expanding the data to multiple species. The reason is doing this way, doing this kind of large scale data integration, we can do it and compare. If we just doing the lab based work, we don't need to compare. We just probably identify the gene and testing the function, doing the PCR, probably follow up some other stuff. But when we're doing the computational jobs, or either way we design the computational based research projects, we are more like to collecting, or either we say intent, or we say interacting, integrating multiple dimensional data for the large scale compare. In this kind of compare, we can find a lot of the mislinks. So for example, in this small projects, we build a LGDB. It is a database. It is a bioinformatics database. We focus on all the red algae genome available in the public. We're collecting the genome assembling. We're doing the annotations. We predict the genes, and we also annotate those genes. What's the function for those genes? Like some of genes, they are relevant for the lipid metabolites. So we are doing those kind of annotation in a consistent way. So this is one of my PhD research projects, and we say this is very useful to helping our local biologists to answer a lot of questions. Like they build the phylogenetics trees, like they collecting the transforming factors, identify the transforming factor targets, something like that. And we say there's a lot of this kind of small needs from our local research communities. And this is the way we are thinking about. We're probably going to expanding our LGDB for large scale data analysis. So this is the web interface in the left side. So this is our web interface for our LGDB. Inside of LGDB now, we have been collected 97 red algae genome assemblies. And then 73, we have been annotated them. And we doing the comprehensive annotation for the gene ontology, protein family, and also the transforming factor binding. So this is very kind of useful for us doing the large scale data integrations. But the LGDB is just a starting point for my talk today. So like the LGDB, they have been providing us the comprehensive search. We can search the annotation data, and we can do in the BLAST. So the BLAST means we, like for example here, we providing a sequence, either way it's a protein or DNA, and they can search against all the data inside of LGDB to find the similar sequence. Based on this kind of way, we can figure out in which red algae they have similar proteins, can implementing the similar biological functions. And we also implementing a lot of dynamic data visualization features. So this is our LGDB. So this database we have been submitted to, we are still in the revision stage, so not published yet. So now we have a well annotated bioinformatics database. So what's the next? We are trying to converting those kind of data to the large scale, to build the large scale data analysis workflow. So for example, we can search the sequence, but researchers, like the biologists, they're not just stop at the sequence. Like they probably prefer, after the BLAST results, they would prefer a summary for what's the multiple alignments for those proteins from different species. They probably prefer to build a phylogenetics trees to find out what's the evolutionary history for those proteins, returned from the BLAST search. And those kind of things, if every request from our collaborators, you probably need to spend 1 or 2 days, to dig into what kind of tools you are going to use. And do we have enough data? Do we need to download new data? Do we need to set up the new systems? So it's very time consuming and it's very hard to reproduce the same results. So this is the reason we are thinking about how can we make this processing, the data analysis, more reliable, more automatic. So this slide just highlights what kind of the analysis can be followed up by the BLAST search. So the BLAST search is just search the protein or DNA sequence against a background database. The background database could be all the known protein in this world, or either way just a highly annotated protein, or either way we just specify one species, all the protein from 1 or 2 species. And then follow up, you can say we can run in the phylogenetics analysis. We can infer the evolutionary analysis. We can infer the evolutionary relationships. For those kind of jobs, they probably ask for moderate E-value. E-value is one of the key cutoff used in the BLAST search to help us filtering the results. And they will be asked high maximum hints, and will be also asked for the taxonomic diversities, balanced clades. Normally, the typical follow-up bioinformatics tools will be something like IQ trees, fast trees, and also after BLAST search, we can doing the functional analysis. For example, we can doing the BLAST to go, and then we can annotating those kind of proteins with the gene ontology functions. We can also doing the interpro scan to identify the domain functional domain inside of proteins. So there are tons of this kind of different follow-up analysis after we're doing the BLAST search, like the pathway, Oslo screening, motif discovery, and structured template search, and comparative genomic search. Huge number of these things, and you will say they have different criteria for the BLAST. Like they probably use different criteria when we set up the BLAST search. And also they have different follow-up BLAST. They have different follow-up bioinformatics tools. So this is kind of the way we can think about how can we use the AI to help us doing the automatically routing different jobs to different tools. So this is why we need to setting up the AI agents to helping us doing the decisions and to run in the systems. Next slides. So if you have been heard about maybe in the last 2 months, this one of the big stuff is it's a tools called OpenClaw. This is an open sourced purely AI agent-based tools. They can run locally in your local computers. They can help you reply the email. They can help you set up the calendars. They can help you probably buy the stocks and sell the stocks. They can do a lot of things if you give them the privilege to doing that. This is something we call the AI agents. Normally, what they do is they have a lot of the user inputs and they understanding the user ideas, and then they connecting with some of the remote AI agents, AI systems, and then they probably also need to use some of the local database or local files, and then combine all those kind of things together, response to the inputs from the user. They are not just like you use the ChatGPT, you ask a question, they answer your question. You probably ask them like saying, book flight tickets from Melbourne to Sydney. Then they can make the booking for you, make the payments for you. If you allow them to like control your bank accounts, if you allow them to control your web browser, they can do these things for you. It's not just a stop at, okay, tell you the price from Sydney to Melbourne is just $200. No, they are going to make the real booking for you. I know there's a lot of the potential, like the ethic, probably safety problem, behind this. But for our research, we are not going to doing this kind of like the connecting with your financial bank, financial accounts. We're just doing the research. We are trying to use the similar ideas to doing automation for our bioinformatics analysis. So you will say we have large scale analysis. So this is the needs we have. And our users, normally they would like a lot of actionable insights and the follow-up analysis based on our BLAST search. So the AI agent is kind of the best way, can fit in these needs for the users, can helping us build those kind of system. Smart, they can run in the follow-up analysis in a smart way. So this is why the LGDB project now is evolved from a database to an analysis platform. We're starting from the LGDB. I'm going to introduce one of the system we call Smart Blast. So they can add in the advanced analysis after. So based on the LGDB, because we are still developing these tools, they can't do everything, but at least they offer a kind of the solutions for our collaborators to help in to doing some of the simple follow-up analysis. Okay, so let me just show you. So inside of our BLAST search, one of the big problem is what kind of background, what kind of reference you are going to use for your analysis. So normally we probably use the NR database. That means a non-redundant database. They include all the proteins or either DNAs from all the species on this earth. For the NR-based BLAST search, you can return very comprehensive BLAST hit, so BLAST mapping. But if you choose, like for example, as I shared with before, like for LGDB, we just included 97 red algae genome. If we use the LGDB as the reference for the BLAST search, then the BLAST mapping will be only focused on all the proteins or DNAs inside of our LGDB. That means 97 red algae genome or proteins. So we use the LGDB as a default reference list for our smart BLAST. The reason is when we're doing the BLAST search, it can only tell you which proteins similar with your input proteins, but they can't tell you what is the gene ontology annotation for the mapped protein. But if in your database, for example, in our LGDB, we already have the annotation information. This can make us follow up the BLAST. I can make the summary for the gene ontology annotation automatically. So this is one of the way you can say if you have a good and comprehensive, well-annotated bioinformatics database, BLAST can helping us doing a tons of the follow-up, high-quality, reliable analysis. So this is just the web interface. I will show you later. So this is our web interface. We just drag the like the FASTA file here, and then we can pick up what kind of analysis we are going to run in, and then we can pick up what kind of database we are going to run in against, like NR database or probably LGDB or Swiss Pro or probably PDB if you are interested in running in compare the 3D structure based data. So we let user to input their sequence. And then we doing the BLAST, and then we can doing the BLAST search. Then we can generating a lot of the commands for the large scale data analysis. I'll show you guys the real systems. So this is the web interface. Now this is just running in my local. So this is the web interface. We drag a protein sequence here, and then we pick up the rights analysis. Like we have probably a lot of different analysis options. We pick up the most the most popular ones, for example, the functional notation, and you can put some of the like the prompt here, or either way you just use the default one, and then AI can help you doing the decision. Now we can pick up which database you are going to run in the BLAST against. Like we just against the local LGDB, and we can also specify one or two species inside of our LGDB. So we have different choice. We just leave its default because we pick up the functional notations. So that's why the system helping us set up the e-value, set up the maximum hint automatically. But you can change those kind of things. After we uploading the data, set up those kind of prompts, and then they can generate in some of the codes for us to do in the analysis. This is just a code for the analysis, but they are not really running this into an actions. As the first stage, my idea is, so the reason for me doing this job is in the first stage is just generating large scale commands for the data analysis. So for example, if my collaborator have an inquiry about, I have 10 proteins, can you help me run in the BLAST search and find out what's the gene ontology enriched in these proteins? And then might generate saying I probably starting running the BLAST and then trying to make a code, trying to transfer the gene ontology notation from the mapped results. But this is very time consuming. Every time I need to doing the same thing, But now I just use the AI to helping me doing this job in a consistent way. I know you probably don't understand this code. This is kind of code I can reuse for any kind of similar analysis. Every time just the AI generated for me, and then I will be going to review this code. If this is properly, if this is what I need, I just copy this code and run it locally. So this is the way in my first stage, I just trying to generating some of scripts based on different scenarios. For example, so this is the functional notations. If we change another one, final genetics analysis, we can do in the generating the scripts. So you say they generating another scripts. This script is for the final genetics analysis. So in this way, at least they save my time. I don't need to every time for the small task. I need to just repeat the similar jobs, change the file name, doing this kind of set up the similar
So sorry, I
computation
mean,
environments.
just being considering of the time, just so you know, yeah.
Sure. Will be quick. Yeah, so this is the first stage. I just generating the code. And then next one is I probably from this generated code, I can doing a follow-up analysis. I probably doing a demonstration for you guys. So for example, we have been doing this final genetics trees, and then we can start in the follow-up analysis. So this is the real time you can see in here. This is the real time they are running the final genetics analysis, running the blast search, and they are going to generating the final genetics tree based on the blast search. So this is the way we can use this system. So our smart blast system can do in the final genetics analysis, also doing the function notation, or also we can do in the pathway mapping, or probably if you are interested in the homolog mapping. So all these kind of things, you can use our systems, can helping us doing this, so doing the jobs in an automatic way. So this is kind of analysis we can do. We can do in different functional and the follow-up analysis. And the beautiful design for our tools is a lot of the follow-up analysis. They probably reduce a lot of the tools. For example, inside of our systems, we have very good design sequence parser systems. We have the alignment system. We have the annotation mapping systems. We have the visualization systems and the scoring system. All those things combined together and use different agents to help us assign different workflow. So for us, this system is not just limited to the LGDB. They can apply to any kind of local, high quality, well annotated bioinformatics database. And this is kind of the way we can run in the things in an automatic way. And also we have centralized configuration strategy. We pre-build all the tools database in our systems and save the users to install those kind of tools. And it's simple to set up, just copy paste so just download all the system in one folder, and then they can helping us running those kind of system in a smart way. So in future, we're probably going to upgrading the models and expanding the data to probably running more data. We can handle the large scale data, and probably going to probably going to helping the users can install in the local computers, and we probably share the code and invite more colleagues and people interested in our systems. We can build the system together. Thank you so much. All good, Max.
Thank you very much, Min. So yeah, well done. And this would have been really useful a few years ago for me. Still useful now. Amazing. So like, do you have funding to do this, or you are developing this on your own at the moment?
It's just for fun. There's no
Okay.
funding, so that's
Yeah.
why this is just the earlier stage, but I can say it's working. So that's why And so I probably publish the code and trying to think about if we can find more collaborators and probably apply the findings.
Oh, great. Has anyone else any questions to Min?
Yeah, Min, are you planning to have an API so you could have like your local agent and then it can interface with the LGDB using an
Yeah,
API?
that's what I mean. One of the things, I mean, for use the AI, one of the things is, yeah, now I just use the API from the ChatGPT. So it's more convenient, it's more smart. I would say running a local large scale language model probably I mean, it's kind of balanced. If we are going to run in a large scale, definitely we will save a lot of money. But if in just like the daily use, like most of the time, we just small jobs from our collaborators. And this is a quick idea. Doesn't matter. We just probably just use the OpenAI API will be fine.
Okay, I see. Thanks.
Hi,
Thank
man.
you.
Oh, sorry.
Oh, sorry. Go ahead.
Yeah, that was very interesting. Just checking on the code that it generates. Is that just a pure bash kind of pipeline, or could you set up something like a Nextflow? Also thinking maybe downstream integrated rather than just here's a code, but here's actually maybe a full Singularity instance, so that it's basically just a plug and go, so that it could run
So
on any machine.
yeah, I guess for the first stage, it's just a kind of it's a bash file you can run in. But setting up the computational environment takes a lot of time. So
Mm,
that's why for the first stage, at the very beginning, I just trying to help myself. Can I generate in the code automatically? It can do that. And then I move on. I'm thinking about can I run in this code? So that's will be the second stage you probably seeing. Uh, so they can run a lot of follow-up analysis. They can really generate in the results. So
mm,
it's still developing, but for the code in the first stage that generated, I would say they probably as I'm saying, it depending on your computational environments. You probably in the code, they probably tell you you need to download the NR database from blah blah blah. They also tell you you probably need to install the Blast blah blah blah. You you probably need some of the bioinformatics experience, like running the command line under the Linux, can understanding the code, can follow the code, can set up the code or easily doing some of the troubleshooting to reuse the code. So that's also a reason I doing the second stage, trying to build all these kind of things in our local systems. If anyone they're not familiar with setting up those kind of computational system, they just put all the things in the Linux systems and they can access those kind of things from the web page. And then they can ask and interacting with the data, running the analysis.
yeah. And just very briefly, any connection with the NCBI Smart Blast might be I'll check the links and there just might be a bit of a kerfuffle with names. There is because NCBI has something called Smart Blast. Unfortunately, all the good names get taken very quickly.
True. Yeah, this is a good one.
Thank, Thank you very much, guys. Sorry, we will I keep moving on. And if you have any questions, please contact Min. Thank you very much, Min. This is really exciting. Dealing with omics data, you're going to help a lot of people. I can tell you that. And now, so the talk was more about generating AI to help with our research, but now a completely different talk. Actually, an example of how AI can be used, in aquaculture and in selective breeding, to be specific. And it is my pleasure to present to you Dr. Roy Costilla, a statistician and applied geneticist working across New Zealand's aquaculture sector. And he'll present to us today a comparison of traditional genomic prediction models and machine learning models, highlighting where machine learning offer advantages or not for selective breeding in Chinook salmon. Thank you very much.
Thanks, Maxine, and thanks, me, for the very, very awesome presentation. I'm just going to share my screen now. Please tell me if you can see it. Can you see that? Yeah, can you see that, Max? Awesome.
Yeah,
Thank
all
you.
good.
Thank you. Thanks, Max, for the invitation. I'm a geneticist and applied as a statistician at the Cawthron Institute in New Zealand. And today I'm going to talk about genomic prediction using machine learning in Chinook salmon in New Zealand. This, of course, is not work that I've done on my own. We have lots of collaborators from different institutions around New Zealand. Oops, it's not moving. Okay, so there are three questions that I want to talk about today. The first one is, can machine learning models improve the accuracy of genomic prediction in Chinook salmon? So this is the very first research question I would like to answer. The animal models, the models used in genomic prediction, they are very old models, a bit older than the paper, the foundational paper of AI you presented, Max. The first model dates to the late 1940s with Henderson, a statistician from Cornell University, who developed the first animal model in breeding. But the models actually come, you can trace them to the very origins of biology with Mendel and Darwin, where people start thinking about genes and segregation and the laws of inheritance. So this in terms of statistics, technically, these models have a very, very good foundations, both statistically and in terms of biology. So here we want to see if the machine learning models can improve into the prediction accuracy that we get with the current models in animal breeding. So that's the first research question. The second research question, and it's a promise that these new methods and the new technology hold, it's whether or not these machine learning models can capture non-additive genetic effects. We know that a lot of the effects in the genome are non-linear, non-additive, but the conventional models not necessarily capture this correctly, or the sample sizes that we have, they don't allow to answer that question. So the second research question goes to that are the machine learning model better capture this non-linearity on the genomic association? And the third, but not least important question, is about computational tractability, because you can have a very nice model, very complex, but it's not computational tractable, which means that it won't run in finite time. You will need too much RAM, or you will need too much running time. So are these new machine learning models computational tractable? So that is the third research question. So those are the three research questions we want to answer, hopefully by the end of this talk. First of all, I just want to talk about the place we work. Me and Max, we are based in Nelson, New Zealand, in the South Island, the top of the South, at the Finfish Research Center, which is this building right here. We do research on finfish, but also in algae and many other shellfish species. We are right at the sea near the Tasman Bay, where we get all the water for our breeding. Right. So this is the trial that that I'm going to use the data for. This is a finfish trial with salmon, with Chinook salmon, that were brought to the Cawthron Institute for a thermo challenge. You can see here we have a few tanks where all the controls, all the conditions are very controlled: temperature, oxygen levels, pressure, and you can see a few of the fish in the trial. These are Chinook salmon, which is, they are also called Pacific salmon or king salmon. And they are different to Atlantic salmon because these salmon are originally from the Pacific coast, from the California area. And these fish were brought to New Zealand in the late 1800s. And now they established, they are self-sustaining populations around the South Island of New Zealand, but they are also used in aquaculture. And one main interest of the Chinook salmon industry at the moment is how to make this, how to select these fish for the changing environment where they need to be bred for. For instance, this is a sea pen in the Madwara Sounds as well, very close, very in the top of the South Island, where we get more often now heat waves that are changing the temperature so dramatically that increases a lot the stress that the fish are bred, the environments where they bred on. Okay. A little bit of also an introduction into what are we trying to do when we do selective breeding for aquaculture. So the main aim for when we do inbreeding in aquaculture is select animals with superior genetic merit to enhance the performance, health, or adaptation within a given environment. And here I want to mention something that is very important, that genetics alone can be an assurance of a successful breeding program. To have a successful breeding program, you need genetics, good management, and an appropriate environment. And when I say environment, I mention it's not just the place where they are bred, but also nutrition, disease, veterinary support, and all the things that will nurture that animal. So we have those three pillars in breeding: genetics, environment, and management. The traits that breeders are interested in aquaculture can be broadly separated into three categories: growth and efficiency traits, fillet quality, and disease resistance and welfare. So how does the breeding cycle works in general? So first of all, and this is an schematic representation from a paper from 2023. We have to decide which are the traits that are important for us, for the breeder. They could be either farmer-driven or consumer-driven. And then we start measuring for those traits. So we do what we call phenotyping for those target traits. It can be either heat tolerance, protein quality, yield, pest, or disease resistance. And that this doesn't apply. This applies to many other species, not just aquaculture species. So once we do this recording of the target traits, we can start selection for parents. And one powerful method that we've been able to use in about since 2010 onwards, it's a genomic selection. I will tell you more about it soon, but basically here we go into the genome of the target species and start mapping the loci that are important for a particular trait. So once we generate our genomic breeding values for our selection parents or for selection candidates, we can start doing our crossing and mate allocation, right? And then we get new varieties, new cultivars, or new germplasm in fish, in plants, in cattle. Applies to any farm species. And how do we do it in aquaculture? Genomic selection. Well, we go and select samples, biological samples from the fish. This is here. We're taking samples, tissue sample units from the adipose tail or from the tail of the fish. We take this into the lab, into the machines, do the sequencing or genotyping. And then we get the genomic data back from the lab. This genomic data is not only genotypes, SNPs, single nucleotide polymorphisms, but also it can be any other genomic data like SNBs or short insertion deletions. So how does the genomic selection work? We have a reference population where we have measured both the phenotypes and the genotypes. We derive our prediction equation for this reference population. And then we have some selection candidates that we don't know their phenotype or we don't know their genetic merit, and we're going to predict their genetic merit using this prediction equation. And from those, we get our selected breeders that we will continue for breed for the next generation. One important thing about genomic selection is that the genomic effects are typically very, very, very small, approximately 2 to 3, maybe more times smaller, orders of magnitude smaller than the phenotype scale. So that means that to estimate those genomic effects, we need very, very large data sets. Here, this is a classic paper in quantitative genetics from 2009, Goddard and Hayes. They calculated theoretically the number of individuals in a reference population for different trait heritabilities. So for instance, for a reference population with 2,000 individuals, we will get accuracies going from about 0.2 if we have a very low heritability of 10%, and that accuracy will go about a bit higher than 0.5 if our heritability is 80%. And you can see if we want to get very high accuracies of around 0.8, we will need very, very high, very big reference populations for a heritability, for the highest heritability, for heritability of 30%, we will need about 20,000 individuals in the reference population. So that's very important because it means that if we have small sample sizes, then our genetic effects will have very high uncertainty. How does the genomic selection model work in terms of the model itself? So we first need to predict the SNP effects in the reference population, as I mentioned before. And here we encounter typically the problem of P much higher than N. So the number of variables that we want to predict is much bigger than the number than our sample size. For instance, we sometimes for some species we have about 50,000 SNPs, and we only have recorded about 2,000 samples, 2,000 fish or 2,000 cows. So this model, it's statistically speaking, you can identify those SNPs because you have more SNPs than your samples. So what the geneticists in the past have used, it's the genomic based linear unbiased prediction model, also known as GBLUP, that can deal with this P higher than N problem. So in terms of algebra, this is the only question I'm going to present today. Sorry, I'm a statistician, so I have to present equations. This is the GBLUP model in terms of algebra. Here we have a vector of phenotypes. We have some fixed effects, but this is the important term here, the set A, where we have the genetic effects. And we estimate these genetic effects using, consider them as a random effect. So here we construct the genomic relationship metrics between all our samples, all the fish or all the individuals in our sample. and estimate the genetic additive variance that is associated with that genotype matrix. So this is in a nutshell the GBLAB model in animal breeding. Another important thing to consider when we're talking about genomic selection is that we usually are dealing with what in quantitative genetics they call complex traits. What are complex traits? So we see genetic variation for most traits in animal and plant species, and this includes aquaculture. But how is that genetic variation generated? Well, it could be several ways. One way will be a Mendelian, polygenic, or infinitesimal inheritance. So Mendelian traits are traits that are controlled by a single or very few number of genes. Polygenic when we have more genes, and infinitesimal when we have a very large number of genes. So in other words, in simple terms, is it this trait is controlled by one gene or many? And here we have two examples from poultry and plants. So the infinitesimal model assumes that the traits are determined by an infinite number of unlinked and additive genetic loci, each with an infinitesimal effect. So in other words, all the regions in the genome have an effect in the trait. This is the foundational model of animal breeding theory. And it's been very useful in practice. Like, for instance, in the selection for chickens, breeding has been able to decrease the time to market for broiler chickens from 16 to 5 weeks in 30 years. So it's almost a fourfold reduction in the time that you need to breed this animal for. But the infinitesimal model is useful, but we know that there is, even though they might be big, there's a finite amount of genetic material. We know that there's a finite number of genes and enhancers in the genome, however big they might be. So the question is then, what is the distribution of effects for a typical quantitative trait? And here we have a couple of examples. I just selected a couple of examples. This is an early maturation in Atlantic salmon. The Loki strain of Atlantic salmon is a recent paper from 2026. Here, the authors use about 700 samples and almost 50,000 SNPs, and they found very clear evidence for two genomic loci that were significantly associated with early maturation in these species. So that's one example. So this trait, early maturation, for this species seem to be controlled by only 2 QTL. But we have very different numbers, like another trait. This is resistance to P. salmonis, which is a bacteria in rainbow trout that also affects salmon species. Here, the authors use almost 500,000 whole genome sequence imputed variants for over 2,000 samples, and they found way more QTLs than other authors. So in this case, they found 5 QTLs all around the genome, and all of them in different chromosomes. So here we see it's a little bit of an egg and a chicken problem, because we have models that have some assumptions, but we don't know if they hold, but we feed the data. And after that, we check whether or not those assumptions hold. So in general, there is no hard rules. Every species and every different group of traits, they can have different heritabilities, from going from Mendelian to polygenic or to the infinitesimal. Okay, so now moving into salmon. So this Chinook salmon trial that the Cawthron Institute ran a couple of years ago it's a thermo challenge trial, but I'm just going to analyze the traits at the beginning, before the actually thermo challenge started. So these traits are weight, fork length, and specific growth rate. I'm gonna the type of genotype data that we use is GBS data, genotyping by sequencing, also known as RAC sequencing data. This data has we apply very high quality filters. In particular, we use only SNPs with a depth rate more than 2, so we can differentiate between the homozygous and heterozygous calls, and a minor allele frequency of 0.05. We also imputed this data just to make sure that any difference between the animal breeding models and the machine learning models is not due to the imputation method. You could also do it without it, but we just wanted to compare like with like exactly. And the genomic prediction models I'm going to be using are GBLAB, the model I just described before, BayC, which is also a Bayesian model using animal breeding, Bayesian neural networks, and extreme gradient boosting, also known as XGBoost. This is the data, the number of animals and genomic loci we're going to be using. In the end, we use just about over 5,000 samples, 5,000 data from 5,000 fish, or just over 9,000 SNPs across the Chinook salmon genome. This data has about a call rate of 90%, which means that it will have about 10% missing, which is not much at all. And as I mentioned,
sorry, Roy, just be conscious of the time.
Yep.
Yeah.
So 3 minutes, 5 minutes. Okay. The data has a mean sample data of about 3. The validation is a tenfold cross validation between the training and the validation sets. So we repeat that 10 times. And the measure of accuracy, it's the correlation between our predicted breeding values and the adjusted phenotypes. So these are the results. On the x-axis, we have the accuracies achieved by each different model on average. And on the x-axis, we have all the different methods that we tried. So you see both, all methods have very similar accuracies, a bit higher between GBLAB and BayC, so about 0.53, and a little bit lower for the neural networks and the XGBoost. Although I have to say not much lower than that, they probably about 0.5 and 0.49. So not very different. It's slightly different, but equivalent. So the interesting thing here is then what have we learned and what are the limitation and advantages of using the machine learning models? So the first one is that these machine learning models only have only are univariate, so they just run trait by trait. They require a lot of tuning of the hyperparameters. So you spend a lot of the computational time just finding those optimal parameters in a search grid. There is no uncertainty around the predictions. So these are I would say the main limitations of these machine learning models I tested. Advantages, they have very similar results without any assumption about the genetic architecture. So they didn't assume that we have the infinitesimal model, or the trait is Mendelian, or the trait is polygenic. They are assumption free. So that's really cool. That's really awesome. And they also don't need imputation for the features. You don't need necessarily to use the genotype imputation to use the method. Just to conclude, these results that I just presented are actually what others found as well. This is a study from 2022 that did over 200 comparisons from 34 studies and found very similar results. The best methods were either the reproducing kernel Hilbert space regression or Bayesian methods, and so it wasn't the machine learning methods who were topping all the others. Also, others have found similar conclusions. Gianola in 2002 found that some machine learning models procedures are sometimes better, but often worse than either BLAB or other members of the Bayesian alphabet. And the same with Ben Hayes and his group. They also found that BLAB much of outperformed, with rare exceptions, machine learning methods in over 200 studies. So the conclusions, just to finish off, to answer the research questions we had at the beginning, can machine learnings improve the accuracy of genomic prediction? Not yet, but watch the space. Are they able to better capture non-additive genetic effects? The same, not yet. Are they computationally tractable? Yes. For this current data set, which is about 5,000 fish with 9,000 SNPs, yes, they run in a tractable manner within less than an hour. So that's something very positive. What is next? For us to try richer, bigger training data sets. What we've used now are genotypes in the form of a non-reference allele count. So a big matrix, N times P matrix of 0, 1, 2s. But this data is actually coming from a sequencer where you have sequence rings aligned to certain positions in the genome. So then the richness of this data set is much, much higher because you have 0, 1, 2, and up to potentially a very, very, very large number of reads per genomic loci. But we're reducing this data into 0, 1, 2. So maybe if we feed directly the sequence read, that will help with the prediction. The other one to try is to use a much larger number of features. Like for instance, in the current genomic evaluations for the Chinook salmon, we use about 40,000 SNPs, and we can also use way more than that if we're using whole genome sequence data. The other next step is the data integration with multi-omic data, gene expression, and methylation. With that, I just want to conclude and thanks everyone that is involved in this research, New Zealand King Salmon, the Ag Research Group of the Bioscience Institute, Victoria University, and the Ministry of Business and Innovation and Employment in New Zealand. Thank you.
Thank you very, very much, Roy. I'm sorry, it will be a bit short in question, maybe only one question. No? Okay, feel free to contact Roy if you have any. Thank you so much, Roy. And so now to our next talk, actually a really interesting fact. Microalgae, for example, now we've gathered more and more knowledge about them. But actually, I've recently learned, thanks to David, that although we know that about their proteome, 65% of this proteome is actually dark. What does this mean? So actually, we know that there is protein there, but we do not know what they necessarily are, how we can classify them, and their function. And actually, improving protein classification then could improve our biological knowledge. And while improving biological understanding, there is also the potential of discovering proteins with interesting characteristic and therefore biotech potential. And as that's why what David has developed with his team is truly fascinating. So Dr. David Nelson is a senior research scientist at York University in Abu Dhabi, and he uses cutting-edge AI to understand how microalgal genomics can inform planetary primary production and environment-responsive producer distribution pattern. So today we'll show us how protein large language model can shed light on the microalgal dark proteomes by achieving near complete protein classification. And importantly, we'll show us how interpretable language-inspired AI can transform research on non-model organisms. Thank you so much. The floor is yours, David.
Great, thanks, Max. Thanks a lot. Thanks for the invitation. And thanks to Roy and Min for their great presentations as well. Very interesting stuff going on. All right, I can share my screen. Share. Yeah. All
Yeah,
right, can you see it? You got
all
it?
good.
Okay.
Yeah.
Yeah, like like Max said, I'm working at NYU Abu Dhabi right now. Here's our campus at night. It's a very nice campus. We are on basically a subtropical island. It's like a desert paradise. It's very nice. And I should note that I'm working in the Laboratory of Algal Artificial Intelligence, Synthetic and Systems Biology. We do all of that kind of work, the A2S2 group. And the PI, my boss, is Kurash Salehi Ashtiani. I'll talk more about some of the work he's specifically doing. Okay, my talk's 4 parts. First, I'll talk about predicting.
Sorry, we will stop the recording, David, as you.
Oh, yeah. Yeah, thanks.
Yamin, can you stop the recording, please?